You've probably heard about some exciting new anti-aging breakthrough, some molecule that reversed gray hair in mice, or a longevity gene that's all the rage in an exclusive cohort of 92-year-olds from a small alpine village where people eat yogurt, herd goats, and have zero stress beyond how much sun-dried hay they're storing. It's inspiring, sure. But here's the awkward question no one seems to ask. Whose aging are we actually studying? And more importantly, does this data apply to everyone else, like you, me, or anyone who isn't living a blissfully unprocessed life at 5,000 feet above sea level? The unfortunate truth is, anti-aging science isn't very global. The field is largely dominated by research from North America, Europe, and a handful of wealthy Asian countries. The cohorts used to study aging, especially in the context of high-tech biomarkers like epigenetic clocks or microbiome signatures, are often composed of people who are white, relatively affluent, and living in stable environments with access to advanced healthcare. This isn't just a demographic footnote. It's a scientific limitation that raises uncomfortable but necessary questions about how applicable our current understanding of aging truly is. Let's take a classic example, the Dunedin study, which helped create one of the most widely used aging metrics, Dunedin Pace. It's an incredibly thorough, decades-long study. But it's also based entirely on people from one town in New Zealand, most of whom are of European descent. That's great for internal consistency, but it's hardly the United Nations of biology. Similarly, GrimAge and PhenoAge, two other popular epigenetic clocks, were calibrated mostly on datasets from US and Swiss populations. This is a bit like designing shoes for the whole world based on the average foot size in Geneva, technically precise, but culturally and biologically narrow. So, why does this matter? Because biology doesn't operate in a vacuum. Ethnicity, geography, diet, pollution exposure, stress levels, and even gut microbes vary wildly across populations, and all of these influence how we age. A calorie-restricted diet that extends life in suburban Boston might have wildly different effects in urban Lagos or rural Indonesia. The impact of omega-3s, intermittent fasting, or rapamycin might be moderated, or even reversed, by genetics or cultural practices we haven't even studied yet. Here's where things get even weirder. Some populations naturally age slower, even without biohacking, supplements, or Silicon Valley-funded cryotherapy. Think about the blue zones, places like Okinawa, Sardinia, or Nicoya, where people live longer not because they've sequenced their methylome but because they eat beans, walk a lot, nap when they feel like it, and live in multi-generational households. No one's injecting NMN in Korea. Yet they have lower rates of chronic disease and dementia than most cities with 5 Whole Foods locations. Unfortunately, many of these regions are understudied, and even when they're examined, the research tends to focus on what they eat or how often they walk, without diving into molecular aging markers. The result? A heavily skewed field where interventions are developed based on narrow data but marketed as universally effective. It's a bit like trying to fix every car on the planet using only the blueprint of a Volvo. Now, let's talk microbiomes. The gut, hailed as the second brain, plays a huge role in inflammation, immunity, and aging. But here's the kicker. Your microbiome is shaped by your diet, your geography, your water, your pets, your dirt, and your traditions. A Western-trained researcher may find one optimal microbial profile based on people who eat sourdough and kale, but miss entirely the healthy aging of a community thriving on fermented cassava or seaweed and pickled plums. Even environmental exposure is wildly unequal. People living in industrial zones age differently than people in pristine coastal villages. Air pollution, access to green space, UV radiation, stress levels, all of these interact with genes and epigenetics in ways that science is still unpacking. So when we read a headline about some molecule that slowed aging in humans, it's worth asking, which humans? Because if the sample population is 90% Swiss retirees living in mountain air, and the other 10% are lab mice, we might not be capturing the full story. To be clear, this isn't a call to throw out the science we have. It's about expanding and diversifying it fast. We need longitudinal studies that include African, Latin American, Middle Eastern, Indigenous, and Southeast Asian populations. We need clinical trials that test interventions across genetic backgrounds and lifestyles, not just across BMI ranges and cholesterol levels. We need to invest in global biobanks and make sure that cutting-edge tools like epigenetic clocks are recalibrated for different populations. If we want anti-aging interventions to be real, they can't just work for people with personal trainers and Nordic bloodlines. In the end, longevity science should aspire to more than just longer life. It should aim for equitable, inclusive, and truly global healthy aging. Because until we start treating aging as the diverse biological experience it is, we'll keep producing solutions that work beautifully for a very narrow slice of humanity. The rest of us? We'll still be aging the old-fashioned way, faster, harder, and far less informed than we deserve.